Now let's see question number 51. For the following, now these are assertion and reason based questions. Now all of you must be familiar with the change in the Ames pattern. This is typically a question of new pattern of Ames. All of you must be knowing that assertion and reasoning questions will be based कि whatever assertion they have given, whether that reason is explaining it or not. Now let's switch over to question number 51. Here you see in electrophoresis, a protein will move towards cathode if the pi is below isoelectric point, and the reason to this is the protein has a net negative charge and so it will migrate towards the cathode. Now before undergoing the explanation of this question, you should have a knowledge of about isoelectric point. Now all of you know that amino acids have a zwitterine structure. Zwitterine structure means they have on one hand amino group and on the other hand they have one carboxylic group. Means they can behave as acids as well as a base, and that is why they are also known as the amphoteric substances. Now this is basically the pi of amino acid on which the net charges are equal, mean positive charge is equivalent to the negative charge or net charge will be 0. If we take the acidic side, what will happen? It will have this type of structure, positive H, CO neg H and H positive, and on the alkaline side it will have the structure and R in general. So now see, this is what decreased pH, that is acidic medium. This is what increased pH, that is alkaline medium. Now what happens is if pH is less than pi, the net charge, as you can see, it would be positive on a protein. On the other hand, if pH is more than pi, the protein will have a negative charge. Now if protein has a positive charge, it is going to move towards cathode, which is negatively charged. On the other hand, if it is negatively charged, obviously it will move towards anode, which is having positive charge. As you know कि cathode being negatively charged, only the cations which are positively charged, they will move there. Now the question says if pi is less than pH, that means assertion is what? In electrophoresis, a protein will move towards cathode if the pH or pi is below isoelectric point. This is correct. It is going to move towards cathode. On the other hand, the protein has a net negative charge and so will migrate towards the cathode. Now see, if a protein has a net negative charge, that means pH is what? pH and pi mein there is a difference. pH is more than pi, and instead of moving towards the cathode, it is going to move towards anode. So the answer to this question should be assertion is absolutely correct, but the reason is false. Or you can say it would be C. Assertion is independently true, correct statement, but the reason is independently a false statement. So we can say whenever pH is less than pi, obviously it is like the movement is like this, and when it is more than this, the movement is like this. And this is what is the zwitterine structure when the net charge is 0, which is also known as neutral pH or isoelectric point. At this point we can separate a particular amino acid, and every amino acid has a specific pi of its own. Let's move to question number 52. epigenetics is? Now see, it is clear from the word itself. Epi means above, upon genetics. The options are, basically epigenetics is simple chemical modification of DNA, because DNA is basically transcribing into RNA and RNA is translating the information into proteins. So epigenetics is, there is no change in the sequence, but yes, the modification, it will be altered, it will be reversible variation, means they are altering the expression of DNA. Now how it undergoes is, epigenetics ke liye there are two processes basically. This is basically a post-replicational modification. What can happen is, DNA can undergo methylation. Means methylation means attachment of a methyl group or on DNA, so that either it can increase the rate of transcription. This methylation is done in the presence of methyl transferases enzyme. A very good example of DNA methylation is CpG islands. Second example is histone acetylation. In the previous question, we have already discussed this histone acetylation in detail. Histone acetylation means they are acetylating the residues so that DNA comes out of the vicinity of the histones and it can activate transcription. On the other hand, if deacylation is there, it is going to decrease the rate of transcription. These are all the examples of epigenetics. Epi means upon genetics. There is a chemical modification of DNA by either acetylating or deacetylating the basic amino acids, that are the histones, or methylating the DNA helix. Then we switch over to question number 53. Now see the following graph. This is a typical graph of effect of substrate concentration on the activity of the enzyme. Now what happens is, enzyme activity it is clear from the word. How active an enzyme is means how active that enzyme can combine with the substrate, forming enzyme substrate complex, which dissociates to form the enzyme and the product. How actively this reaction goes. Now experimentally, what are the factors which can affect the activity of the enzyme? pH, temperature, and most important is substrate concentration. Because we cannot verify enzyme substrate complex, we cannot experimentally judge ki how much enzyme substrate complex is produced. For this, there were two scientists, Michaelis and Menton. They gave an equation to explain the kinetics of the enzyme, and the equation given by Michaelis-Menten is V0 is equal to Vmax S over Km plus S, where V0 is the initial velocity, Vmax is the maximum velocity, S is the substrate concentration, and Km is the rate of association as well as rate of dissociation. Means if this is K1, rate of association to form ES, K minus 1 undissociated ES, K2 product formed, and K minus 2 is again the ES which is left over, which is not converted into product. The sum of all these four association and dissociation constants is Km, which is Michaelis-Menten constant. Right. Now let's see how this pH, how this temperature, and how this substrate concentration alter the enzyme activity. If we talk about pH, every enzyme has a optimum pH. However, there are certain exceptions that alkaline phosphatase works at pH 11. Pepsin works at pH 2. But overall, if we see, all the enzymes have a pH optima between 5 to 8. Similarly, temperature, enzymes work best at 35 to 40 degrees. If we increase the temperature, if we decrease the temperature, what will happen? Enzyme will denature, because it is nothing but a protein. And if we see the graph in pH and temperature, it is bell shaped. It is like this. This is a bell shaped graph in pH and temperature. But in substrate concentration, since substrate concentration plays a very, very important role, what happens is it has a hyperbolic graph or curve, hyperbolic. Now see, what will happen is enzymes take the substrate at the active site. So in the starting, when the reaction has just started, initial velocity mein there will be what? More the enzyme, more will be the substrate concentration. But a stage will come when all the active sites of the enzyme will be occupied by the substrate. At this particular point, it is going to attain a saturation state. This is also known as zero order kinetics. This is also known as saturation kinetics. And this is also known as steady state kinetics. State kinetics. Now see, why zero order? Because now enzyme and substrate are independent of each other. They cannot bind. Why saturation? Because saturation has been attained, and that is why it attains a steady state graph. And always remember, when initial velocity is half the maximum velocity, this Km is equivalent to the substrate concentration somewhat here. So when V0 is equal to half Vmax, Km is equivalent to the substrate concentration. And the significance of Km is it depicts the affinity of the enzyme with the substrate. If we say it is basically inversely proportional, if an enzyme has high Km, that means the affinity of the enzyme with the substrate is low. Similarly, if it has low Km, affinity of the enzyme with the substrate is high. That is what exactly happens in case of hexokinase and glucokinase. Hexokinase can work even when the concentration of glucose is too less, whereas glucokinase will work only and only when the concentration of glucose exceeds 100 milligram per deciliter. So this is what is zero order kinetics or steady state kinetics, or effect of the substrate concentration on the enzyme catalyzed reaction. Now see, look at the question. Question says the curve is hyperbolic in nature. Obviously it is a hyperbolic graph. Question mark here represents the Km of the enzyme. Obviously this represents the Km of the enzyme. At point C, only a small amount of the enzyme is present as the enzyme substrate complex. This is wrong, because at point C it has already attained a saturation state. No substrate is left over, as all the active sites of the enzyme have been already occupied by the substrate. That is why it is zero order. And at point C, V is independent of S. So out of these, the answer to this question is option C. Next, question number 54. Pantothenic acid containing coenzyme is involved in. Now see, pantothenic acid is what? The active form of pantothenic acid is coenzyme A. And this is vitamin B5. Vitamin B5 is pantothenic acid, and the active form is coenzyme A. And it basically regulates what? Acetylation. Acetylation. Addition of acetyl group, because what happens is if this coenzyme A combines with acetic acid, it forms acetyl CoA, and if it combines with propionic acid, it forms propionyl CoA. Acetyl coenzyme A is the end product of even chain fatty acids, and propionyl-CoA is the degradative product of odd chain fatty acids. So basically it is regulating what? It is regulating acetylation. Answers are given, options are decarboxylation, dehydrogenation. Now decarboxylation is removal of carbon dioxide. Decarboxylation always requires PLP for its function. Dehydrogenation, NADH is there, FADH2 is there for taking the hydrogen out. Acetylation is carried out by coenzyme A, and carboxylation, biotin is required. So always remember, this is again a very, very important question which has been asked so many times in the exam. For carboxylation, we require biotin. For decarboxylation, we require PLP, which is vitamin B6. It is also required for transamination. Transamination requires B6 or PLP. For dehydrogenation, we will require what? Simple removal of hydrogen, NAD will be converted to NADH, or you can say FAD will be converted into FADH2. So for this, pantothenic acid or vitamin B5 requires acetylation, which is a very, very important precursor of fatty acid oxidation, fatty acid synthesis, cholesterol biosynthesis. Now let's switch over to next question. Question number 55, enzyme deficient in eye cell disease. This is nothing but an example of mucolipidosis 2. And the deficient enzyme here is N-acetyl glucosamine phosphotransferase. And the options are yes, the answer is A. This is an example of mucolipidosis. This is again a lysosomal storage disorder. And all of you know that in this Golgi apparatus is required, because glycosylation, the transfer, basically occurs in the Golgi bodies. So the answer is A. Question number 56, what is the contribution of this scientist? Now you can see this is Hargobind Khurana. And all of you know Hargobind Khurana has a big name in genetics. He got a Nobel Prize also when he was shifted from India. And he was the one who gave the concept of gene also, genetic code. He has played a very important role in the genetic code. So the answer would be this, right? Then we switch over to, answer would be B, not a function of endoplasmic reticulum. Now endoplasmic reticulum, all of you know, it is a very important organelle. It can be smooth, it can be rough. The major difference between rough and smooth endoplasmic reticulum is RER requires ribosomes, so they are basically required for the synthesis of proteins. Smooth means the surface is smooth, that means it is without ribosomes. Now these endoplasmic reticulum has so many functions to perform. In fact, endoplasmic reticulum, Golgi apparatus and ribosomes, these are the protein synthesizing machineries of the cell. Now here not a function, protein synthesis is a function, muscle contraction, protein sorting and glycoproteins. Muscle contraction is not a function of this endoplasmic reticulum. Except this, all are very important. Yeah, endoplasmic reticulum plays a very important role in rest of the three. So the answer here is B. Then question number 58, essay for lipid peroxidation. Now see, lipid peroxidation is what? Lipid peroxidation is this. Rancidity of fat is due to the formation of peroxides in this. We cannot keep fat for a longer time. So there are certain assays by which we can basically see to what extent the fat is rancid, yeah, to what extent that peroxidation has occurred. For this, Fox assay is carried out. Fox assay. And basically what happens is, all of you know that this all depends upon Lambert-Beer's law. Lambert-Beer's law. More the absorbance, more will be the peroxidation. And this is basically evaluated by the formation of the colored products. So here what happens is this Fox is nothing but this is iron, oxidation of ferrous from Fe2 plus to ferric ions in the presence of xylenol. So whatever lipid that has been already degraded, that has been converted into peroxides, it is going to combine, oxidizing this in the presence of this, giving out a chromophore or color. Chromophore or the color. So Fox assay is basically carried out to check the peroxidation or lipid peroxidation or the rancidity of the fat. Others are MTT assay. MTT assay is again, it is basically assessing the metabolic activity of the cell. This is basically a dye, tetrazolonium dye is there, which again oxidizes and reacts with the substance present in that to give out a formazone, which is again a chromophore. So more the cell activity, more will be the color, and obviously more will be the inference. Guthrie's test. Guthrie's test is for phenylketonuria. The next question, end product of purine metabolism in non-primate mammals is. Now whenever you know purines are synthesized by one is salvage pathway and other is de novo pathway. Obviously if purines are produced, they have to be degraded also. The degradative product, whether it is AMP, whether it is GMP, whether it is IMP, the degradative product of all the three is hypoxanthine first. This hypoxanthine will be converted to xanthine, and this xanthine will be converted into uric acid. The enzymes here are xanthine oxidase here and xanthine oxidase here. Now what happens is, in the non-primates, in human beings, uric acid is the end product of purine catabolism. But in non-primates what happens is, this uric acid in the presence of uricase enzyme, the enzyme is uricase, it forms allantoin. But human beings lack this enzyme. So purine degradation completes till the formation of uric acid. But in non-primates, due to the presence of this uricase enzyme, allantoin is the end product of purine catabolism. So here the answer would be allantoin, that is D. And the enzyme deficient is uricase in human beings. Then next, which of the following is correct about the enzyme which has a sigmoidal graph? Now here what we see is, just now we discussed about the effect of the factors on the activity of the enzyme. We took pH, I told you in pH and in temperature it has a bell-shaped graph. And when we consider the effect of the substrate concentration, it is a hyperbolic graph. But whenever we consider allosteric mode of inhibition, allosteric, the graph is sigmoidal. And this is the major difference between allosteric inhibition and the other type of inhibition. See, what happens is, as it is clear from the word itself, allosteric means here the modulator is binding to a site totally different from the active site of the enzyme, so that either it can increase the rate of a reaction or it can decrease the rate of reaction. See for example, if we take here in this graph, we take the concentration of the enzyme with substrate, what happens is it has a hyperbolic graph like this, saturation state, but whenever we have to see the effect of allosteric inhibition, it will have either this type of graph or this. This is what is, this is sigmoidal, and this is what is cooperativity, means if this modulator is activating, this is positive cooperativity, and if it is deactivating, it is negative cooperativity. Binding of heme, binding of oxygen with heme is an example of positive cooperativity. And this allosteric inhibition, the other name of this is feedback inhibition or product inhibition. And the major difference between allosteric inhibition and rest of the inhibition like competitive, non-competitive and uncompetitive is allosteric inhibition never follows Michaelis-Menten kinetics. Always remember this. It will never follow Michaelis-Menten kinetics. And the graph which is obtained in terms of allosteric inhibition, it would be always sigmoidal instead of hyperbolic. So let's look at the options. What are the options? Allosteric modifier binds in a concentration dependent manner. Modifier can affect the catalytic site by binding to the allosteric site. Yes, whenever this modulator, because it is binding to a site altogether different from the active site, so obviously it is going to, enzyme is nothing but a protein, and when a modulator binds, it is going to alter the conformation of that protein. Adding more substrate to the enzyme can displace, and allosteric modifier changes the binding constant. No, so the answer is B. Modifier can affect the catalytic activity by binding to the allosteric site, and it always has a sigmoidal curve and it never follows Michaelis-Menten kinetics. Then question number 61. A girl lick paint that is peeled off from the toys develop acute abdominal pain, tingling sensation of hands and legs and weakness. Which enzyme is inhibited in the child? So it's again a very simple question. The girl is licking the paint, and we all know that this paint consists of lead. So basically this is what? This is lead poisoning. Lead poisoning. Or what happens is this lead is an inhibitor of ALA dehydratase. The second enzyme in the synthesis of heme. Because you remember heme synthesis mein what happens is succinyl coenzyme A combines with glycine to form amino levulinic acid in the presence of enzyme synthase, and this ALA then will undergo dehydration or in the presence of enzyme dehydratase, forming prophylinogen. So what happens is lead is an inhibitor of this enzyme. Lead is inhibiting this. So obviously there will be increased synthesis of this ALA also. Increased synthesis of ALA will be there in urine. And this lead also inhibits ferrochelatase enzyme. Ferrochelatase enzyme is the last enzyme which is synthesizing heme. So here the answer will be ALA dehydratase. D option. Next question is, which of the following is having maximum thermic effect of the food? Maximum thermic effect is simply nothing but the specific dynamic action of the food. Specific dynamic action of the food. Dynamic action, which basically assesses the value above resting metabolic activity. Above resting metabolic activity. And it is highest for proteins. Proteins have 30%, then comes fats, and then comes carbohydrates, 12% and 7%. And that is why whenever we go to gym or whenever we go to a nutritionist, they always suggest us to have a protein rich diet, because they have more that specific dynamic action. So the answer here is proteins. Then question number 63, the following test is performed for which condition? Now this is again excessive hemolysis of the RBCs, and this is hereditary spherocytosis. Now how we can differentiate? By looking at the test tubes. Now see, in first test tube we can see this is a control. Control mein you can see that there is what? This is osmotic fragility test. You can see the lines which are visible, but once the RBCs are hemolyzed, in the last tube, third tube, you cannot see that line, or it is opaque, it is clear. That means whatever RBCs are there, they are hemolyzed. You can compare first test tube occurs as a control test tube, where you can see the lines are visible, and if you compare the first control with the test, it is completely opaque, no line is eligible, no line is visible. So this is what? This is hereditary spherocytosis, and the name of the test is osmotic fragility test. Osmotic fragility test. Let's switch over to question number 64. Regarding LDL receptors, all are true. Now LDL, all of you know, this is formed from VLDL, taking IDL as one of the intermediates. LDL is also known as the bad cholesterol, because its main function is it is helping in taking cholesterol from the liver to the peripheral tissues. Now all are true except it found in clathrin coated pits of, yes, it is found in clathrin coated pits of cell membrane, it is true. Found only in extrahepatic tissue. This is wrong, because LDL is basically formed in the liver, and it is found there in the liver and extra. Its main function is to transport cholesterol from the liver to rest of the tissues. Internalized by endocytosis, this is correct, and high level of cellular cholesterol down-regulate LDL receptors, this is also correct. So out of these four options, the option which is not correct is found only in extrahepatic tissues, because LDL is present in the liver as well as the extrahepatic tissues. Question number 65, the monosaccharide with maximum rate of absorption in intestine is. Now if we compare the rate of absorption of glucose galactose, galactose is 100 times more effectively absorbed than glucose, and fructose and mannose. So obviously maximum rate of absorption is galactose, and this galactose, once it enters inside the cell, it is phosphorylated in the presence of enzyme galactokinase. 1-ATP is utilized to form galactose 1-phosphate. This galactose 1-phosphate then combines with UDP glucose, which is actually coming from glycogenesis. There is interchange, epimerase, because galactose is nothing but it is a C4 epimer of glucose, and it will form glucose 1-phosphate and UDP, galactose will be produced. Deficiency of this enzyme, which is catalyzing the epimerization, will lead to increased concentration of galactose in the blood, causing galactosemia, which is common in infants. And this galactose can be converted into a sugar alcohol, which is dulcitol, and it is having 100 times faster absorption than glucose, and it is basically carried by sodium dependent glucose transporter. Then question number 66. These OSA zones belong to. Now see, what are OSA zones? OSA zones are basically the characteristic features of reducing sugars. All the reducing sugars are going to form the OSA zones. They basically combine with phenyl hydrazine hydrochloride and sodium acetate to form characteristic OSA zones. जैसे glucosa zone is there, glucosa zone. Similarly, fructosa zone is there. Similarly, mannose zone is there. Similarly, lactosa zone and maltosa zone. Because lactose, maltose, even though they are disaccharides, then also they are reducing sugars. Only sucrose will not form a OSA zone. Why? Because it is a non-reducing sugar. Except this, all the sugars will form OSA zones. Now, they form crystals basically. They are kept overnight, and by seeing the shape of the crystals, we can predict which monosaccharide or which disaccharide is forming this type of crystal. So, in glucosa zone, fructosa zone and maltosa zone, they have needle shape. Needle shape, or they resemble the bundle of hay. Lactose has powdery puff and maltose has sunflower petal like crystals. So here you can see, if you see the picture, this is nothing but bundle of hay or needle like. So answer would be D. Glucosa zone, fructosa zone and mannose zone are going to form these type of crystals. Question number 67. Which of the following is not a ribozyme? Now see, we know all enzymes are proteins. Means enzymes have a protein part associated to them. Okay? And if this protein part, just the protein part, is known as the apoenzyme, when it combines with a cofactor or when it combines with a coenzyme, it will become a holoenzyme. But here we are talking about what? Here we are talking about ribozymes. Now ribozyme, as it is clear from the word, in which RNA has a catalytic activity. So wherever RNA is functioning as a catalyst, it will come under the class of ribozymes. Now let's see the options. Transpeptidase, peptide bond is involved. This is what? This is nothing but an enzyme required in translation. This is ribozyme. Ribonuclease, digesting the nucleases, where again RNA is involved, it is a ribozyme. Peptidyl transferase, again peptidyl transferase is a very important enzyme of translation, transfer of a peptide group. Last is poly-A polymerase. Poly-A polymerase is nothing but it is catalyzing the polymerization, means it is adding some residues, yes, attaching some RNA or DNA to it. Here the activity is simple polymerization, so no ribozyme is involved in this. So the answer to this question is poly-A polymerase, which cannot be a ribozyme. Apart from this, all the rest of the enzymes will come under the class ribozymes. The next question, in one carbon metabolism, serine loses which carbon atom? Now see, what do you mean by this one carbon metabolism? One carbon metabolism, as the name suggests, one carbon metabolism means species is giving rise to one carbon. Either it is in the form of methyl group, or it is in the form of methinyl group, or it is in the form of formyl group, or it is in the form of methylene group. What is happening is we are transferring one carbon atom. Right? What are the amino acids and vitamins which are involved in this one carbon metabolism? Most important is methionine. Methionine is required for transmethylation reactions. The active form of methionine is this SAM, and it helps in transferring this methyl group when norepinephrine is converted into epinephrine. This is a very important reaction of methylation. Other vitamin which is required for this is folic acid. This folic acid generates C2 and C8 of purines. This is again one carbon metabolism. Now here in the question it is asked serine. Serine is again one very important amino acid which is donating its methyl group to glycine. Basically it is synthesizing glycine, and the complex here is, it is a reaction. This serine is converted into glycine taking folic acid as one of the intermediate. H4 folate, methyl tetrafolate, and it is also known as glycine synthase complex. Glycine synthase complex. Now the question arises कि this methyl group is present as an alpha carbon atom or a beta carbon atom. Now if we see the structure of serine, serine has this type of structure. This is the basic structure of the amino acid, H, CH2, OH. This is the alpha carbon atom, this is the beta carbon atom. Now serine, if it donates its alpha carbon atom, for example, it won't be an amino acid after that. So there is no question of donating this alpha carbon atom. But if you see this beta carbon atom is altogether free, which can be given to glycine. Even though if it is donating this beta carbon atom, the intact structure of amino acid will be still there. So serine will lose its beta carbon atom to glycine in the presence of folic acid, in the presence of enzyme glycine synthase. It is a complex which requires folic acid as a very, very important coenzyme. The other enzyme can be hydroxymethyl transferase, because the transfer of methyl group is there, which is again one carbon metabolism. So the answer here is serine will donate its beta carbon atom. Then the alpha helix of a protein is most likely to be disrupted if a missense mutation introducing the following amino acid within the alpha helical structure. Now the amino acids are alanine, aspartic acid, tyrosine and glycine. Now out of these, if we see, glycine is the only amino acid which is even though simplest, but whenever it is present in the alpha helix, it is going to disrupt it, because it has a very simple structure. It is optically inactive also. One very important point about this alpha helix is, alpha helix is basically either it is right-handed or it is left-handed. Bonds here are covalent bonds, and along with covalent bonds it has intramolecular hydrogen bonding. Intramolecular hydrogen bonding. Major points about alpha helixes is proline is never found in alpha helix. So one thing, proline never found. Second is if glycine is found, it is again going to disrupt the alpha helix. The reason behind this is it is the simplest one, it has only alpha carbon atom. So bonding with other amino acids would be a little bit difficult. So in alpha helix the answer is glycine. Next question. A chronic alcoholic have low energy production because of thymine deficiency as it is. Now see, whenever we talk about thymine deficiency, the thing which comes up in our mind is thymine, the active form of thymine is TPP, and it is a very important coenzyme of PDH, pyruvate dehydrogenase complex, alpha ketoglutarate dehydrogenase and transketolase. Now what are the reactions catalyzed by these enzymes? Let's see the options first. Acting as a cofactor for alpha ketoglutarate dehydrogenase and pyruvate dehydrogenase, acting as a cofactor for transketolase, interferes with energy production from amino acids and act as a cofactor for oxidation and reduction. So obviously the answer is clear cut A. It acts as a cofactor for alpha ketoglutarate dehydrogenase and PDH. Again I will take these reactions because this is a very important question. Thymine vitamin B1, active form is TPP, constituent of PDH, constituent of alpha ketoglutarate dehydrogenase, and it is a constituent of transketolase. Transketolase is basically an enzyme of HMP shunt. So basically in the erythrocytes, assay of this TPP is carried out. But here we are talking about this reaction. PDH mein what happens is this pyruvic acid is converted to acetyl coenzyme A. Simple decarboxylation is there. And for this decarboxylation only, this TPP is required. So obviously if this TPP is deficient, thymine is deficient, there will be increased concentration of pyruvic acid. This pyruvic acid, instead of being converted into this, will be converted to lactic acid, or there will be lactic acidosis. Similarly, alpha ketoglutarate dehydrogenase is an enzyme of TCA cycle which converts alpha ketoglutaric acid into succinyl coenzyme A. So the answer to this question is, it is a very important cofactor for alpha ketoglutarate dehydrogenase and pyruvate dehydrogenase क्योंकि chronic alcoholic है. It will have a low energy production. The next question, which of the following is true about Krebs cycle? Now Krebs cycle is also known as TCA, tricarboxylic acid. Why? Because first stable product is citric acid which has 3 carboxylic groups in them. That is why it is also known as citric acid cycle. And citric acid is the first product. They were discovered by Sir Hans Krebs, that is why it is known as Krebs cycle. Now, which is true? Pyruvate condenses with oxaloacetate to form citrate. Alpha ketoglutarate is a 5-carbon compound. Oxidative phosphorylation occurs in the cytoplasm only, and Krebs cycle can operate in anaerobic condition. Now see, let's take first option. Pyruvate condenses with oxaloacetate to form citrate. If we see TCA cycle, pyruvic acid is the end product of glycolysis. It is coming from the cytoplasm, entering in the mitochondria, where it forms acetyl-CoA. This is occurring in the matrix of mitochondria. This acetyl coenzyme A condenses with oxaloacetate to form citrate. So automatically first option is waved off. Pyruvate never condenses with oxaloacetate to form citric acid. Now second option is alpha ketoglutarate is a 5 carbon compound. Yes, it has 5 carbon atoms in it. This is one of the major intermediates of TCA cycle, which is converted to succinyl-CoA, then succinic acid, fumaric acid, and then oxaloacetic acid. So this option is correct. It is alpha ketoglutarate is a 5 carbon compound. Next, if you look at the next options, oxidative phosphorylation occurs in the cytoplasm. It is not possible. Whenever we talk about oxidation, means it is involving oxygen. So whenever oxygen is involved, it can never be cytoplasm. It has to be mitochondria. And oxidative phosphorylation is attachment of inorganic phosphate in the mitochondria to form ATP. And last option is Krebs cycle can operate in anaerobic conditions. No. Just now I told you it occurs in the matrix, so obviously oxygen is involved. So the answer to this question is B, alpha ketoglutarate, which is a 5 carbon compound. Next question number 72. A 30-year-old male patient comes to the medicine OPD for swelling which looks like grapes. Now if you see the picture, this is nothing but xanthomas. Right, and this is a characteristic feature of dys-beta lipoproteinemia, which is basically a type 3 hyperlipoproteinemia. The options are familiar this. Obviously the answer would be D, familiar dys-beta lipoproteinemia. Characteristic feature of this is also known as familiar dys-beta lipoproteinemia. The other name of this is remnant removal disease. Remnant removal disease. The lipoproteins which increase in them, lipoproteins which are increasing in this disease are chylomicrons, and VLDL. And obviously cholesterol will also increase. The fat which will increase is cholesterol. And characteristic feature is it forms the xanthomas. They can be tuberoeruptive in the knees, in the buttocks, and they will be in the palms also, which is a palmar xanthoma. So the answer to this question is question number 3, FDBL, dys-beta lipoproteinemia, also known as a remnant removal disease, also known as type 3 hyperlipoproteinemia. So the answer is familiar dys-beta lipoproteinemia, which is D. Next question number 73, pantothenate kinase associated neurodegeneration is. Now pantothenate kinase, pantothenic acid, vitamin B5, deficiency of this leads to Gopalan's burning feet syndrome, and it can also lead to PKAN, pantothenate kinase neurodegeneration. It is also known as Heller-Warden's-Patch syndrome. And always remember this pantothenate kinase deficiency or B5 deficiency leads to Gopalan's burning feet syndrome. Burning feet syndrome. And it is also known as PKAN. Right. So the answer here is Heller-Warden's patch syndrome or B option. The next 74 question, an example of anaplerotic reaction. Now these anaplerotic reactions are basically the filling up reactions. Filling up reactions means what happens is, suppose a sudden influx of pyruvic acid In a normal individual, this is happening. Pyruvic acid is being converted into acetyl-CoA. And normal, this acetyl coenzyme A is combining with oxaloacetate to form citric acid and TCA is undergoing. Anaplerotic means if there is a sudden influx of glucose, there will be increase in the concentration of pyruvic acid. Some of the pyruvic acid will be converted to oxaloacetic acid in the presence of enzyme pyruvate carboxylase. Biotin is the coenzyme. I am mentioning biotin again and again as this is a very important question and it is asked in so many exams. Biotin, which is a coenzyme for carboxylation. Now what will happen is, this is a reaction of gluconeogenesis also. In fact, this is the first step of gluconeogenesis. But this is one of the very important anaplerotic reaction. Conversion of pyruvic acid to oxaloacetic acid. It is required only when there is sudden influx of pyruvic acid. Main part of pyruvic acid will obviously undergo oxidative decarboxylation to form acetyl coenzyme A, but rest of the pyruvic acid will form oxaloacetic acid. And there are so many other fate of oxaloacetic acid also. It can undergo gluconeogenesis, it can form aspartic acid, there are so many other. But here when we talk about anaplerotic reactions, this is the first very important anaplerotic reaction that is given in option B, pyruvate to oxaloacetic acid. Then question number 75, quantitative DNA estimation is done by. Now whenever we have to estimate proteins, whenever we have to estimate carbohydrates, whenever we have to estimate DNA, RNA, it can be qualitative as well as quantitative. But whenever we talk about DNA, it is quantitative estimation, means how much DNA we are taking for different methods. Now quantification is always done with the help of spectrophotometer. This is again very important. Spectrophotometer the working of this spectrophotometer is based on Lambert-Beer's law. Lambert-Beer's law. Basically we study the spectrum. Now suppose if you look at DNA, DNA may you see DNA is double helical in structure, means it has the bases which are linked by the hydrogen bonds, these bases which are linked to sugar by glycosidic bond, and this sugar which is linked to phosphate by again a phosphodiester bond. And all of you know DNA, the strands are anti-parallel. If one is running in the direction 5 prime 3 prime, other would run in the direction 3 prime 5 prime. So if we see the DNA, obviously sugar, phosphate, all will be the same. The difference between two samples of DNA would be what? The bases. A DNA can have maximum GC base pairs, one sample of DNA. Other sample of DNA might have maximum GC base, 80 base pairs. Obviously GC is triple bonded, AT is double bonded, large amount of energy is required to break a DNA which has more GC base pairs, and less amount of energy is required to break DNA which has 80 base pairs. So obviously the DNA having more GC base pairs will give more absorbance, or it will be known as a hyperchromic DNA. Hyperchromic DNA. Similarly, DNA having more AT base pairs, less absorbance, and it will be known as a hypochromic DNA. And always remember, DNA absorbs at 260 nanometers. This is UV, ultraviolet. And whenever DNA absorbs at 260 nanometers, it is basically due to the presence of the bases only. Just now I mentioned whether it is increase in GC base pairs, whether it is increase in AT base pairs. So, and it is measured always by the spectrophotometer. Question number 6, gene editing can be done by various methods like hypermethylation and amplification. Which of the following will not change the genetic code? Now here the answer is clear, it is epigenetics. Rest all the three types, whether it is CRISPR, whether it is Stellan, whether it is Gen Expert, they are all going to change the genetic code. Only epigenetics, it is above genetics, it is basically catalyzing methylation. Just now I mentioned methylation and acetylation. So the answer here is epigenetics, which is again a post-replicative modification of DNA. Question number 77, the given feature is which storage? Obviously this is adrenal calcification of the glands, and this is the characteristic feature of Wolman's disease. The deficient enzyme here is cholesterol ester hydrolase. This is again a very, very important question. Cholesterol ester hydroxylase, and the disease is Wolman's disease. So the answer here is C. Question number 78, real-time PCR is used by PCR, polymerase chain reaction. It is simple exponential amplification of the DNA. Multiplies DNA, multiplication of specific segment, multiplication of protein to know how much amplification has occurred. Answer is clear, how much amplification has occurred, because the amplicons are basically the sequences. For them the primers are designed, and then Taq DNA polymerase is added and the PCR cycle is allowed to run. This is exponential amplification, means 2 raised to power n. If PCR runs for 2 cycles, obviously 2 raised to power 2, definitely these amplicons will be produced. So the answer to this is how much amplification has occurred. This is the basic principle of this real-time PCR, by exponential amplification. Then question number 78, lipoprotein resembles. Now here this lipoprotein, the other name of this lipoprotein is LPA, and it basically has a significance, homology with this plasminogen. It basically interferes in the conversion of plasminogen. So basically what happens is the fibrin clot is not lysed. Clot does not lyses. So the answer to this is A, plasminogen. Then next question, why glucose 6-phosphate in the cytoplasm of hepatocyte is not acted upon by glucose 6-phosphatase as soon as it is formed? Obviously it is why, because the enzyme is present in the smooth endoplasmic reticulum. So glucose 6-phosphate needs to be transported into SCR, where it basically undergoes glucose 6-phosphate enzyme acts and it is converted into glucose. Now options are thermodynamically possible only when gluconeogenesis occurs, need protein kinase for its activation, enzyme is present, yes. So it is, enzyme is present in SCR and glucose 6-phosphate needs to be transported into that SCR. Then 81, identify the inheritance pattern. Now this is again, you have to first of all see, if we look at the inheritance pattern, there are so many different type of pattern of this inheritance. It can be autosomal dominant, it can be autosomal recessive, it can be X-linked dominant, it can be X-linked recessive, it can be mitochondrial inheritance also. Now by seeing the picture is giving us an idea that this male is diseased here, right? And this male, we know ki in human beings it has, males are heterochromatic and females are homogrammatic. Females are carriers and males are generally diseased if one chromosome is having that disease gene. Hollandric inheritance is altogether different, but overall if we talk about the X chromosome. Now here in this picture we can see that the male is transferring the characters to the daughters. Male to male transmission is not seen here because male son is receiving the Y chromosome. So this is what, this is nothing but this is X-linked dominant inheritance. Clear cut. So the answer to this question is option A, X-linked dominant inheritance, whose main features are a father always transfers his characters to the females. Male to male transmission is never seen because the son is having a Y chromosome from the father, and 50% of the progeny is affected. Next question, number 82. Immunoglobulins are glycoproteins. Glycoproteins are which are having protein as well as a carbohydrate. They have a carbohydrate residue also attached to it. So it is glycoprotein. Hormone sensitive lipase acts on. Now the previous question we had discussed about hormone sensitive lipase and lipoprotein lipase. As it is clear from the word itself, hormone sensitive lipase, lipase which is degrading the fats, which is basically degrading the, and fats are present are what? They are nothing but the TGs. The stored triglycerides which are there in the adipose tissues, they will be acted upon by hormone sensitive lipase triggered by glucagon. Because glucagon is activating it, insulin is deactivating it. Insulin inhibits hormone sensitive lipase. In fact, insulin activates lipoprotein lipase. So here it is what? It is simple lipolysis. So hormone sensitive lipase will always act on the triglycerides, breaking them into, again these are TAGs, breaking them into DAGs, diacylglycerols, then breaking them into MAGs, monoacylglycerols, and finally forming free fatty acids and glycerol, which can finally undergo beta oxidation. Sequence shown in the image is present in. Now see, this is what? This is simple the promoter sequences. Promoter sequences present in transcription. Transcription basically is formation of RNA from DNA. First step in this is always initiation, in which the major role is played by RNA polymerase. This RNA polymerase is having 5 subunits in prokaryotes. This is the sigma subunit, and this is alpha dash, beta, beta dash, and omega. This sigma subunit helps in recognition of the promoter sequences. Promoter sequences means from where this transcription has to start, and these are rich in Tata box. Wherever Tata box is there, it is present downstream, and some of the sequences are present upstream. Tata box is known as Pribnow box in prokaryotes, and it is known as a Hogness box in eukaryotes. So this is nothing but a promoter sequence which is initiating the process of transcription. Then finally initiation occurs, then elongation and termination occurs. So this is nothing but promoter region. Next, which of the following is not a part of the fatty acid synthase complex? Now here what we see is, see what happens is, all of us know that fatty acid synthesis is de novo. De novo means from a 2-carbon species we are generating palmitic acid which has 16 carbon atoms. The precursor of fatty acid is acetyl coenzyme A, and it is forming palmitic acid. This is de novo. From 2 carbon we are generating a 16 carbon species. Now first step in lipogenesis is what? This acetyl coenzyme A will undergo carboxylation in the presence of enzyme carboxylase. Again I am mentioning biotin, forming what? Malonyl coenzyme A. Now once this malonyl coenzyme A is produced, the entire function is taken over by the fatty acid synthase complex. This is again an example of multi-enzyme complex. It works like this. These are the different activities which will undergo, and there are the two active sites. One is having cysteine SH and one is having pentotheniine SH. These are the two active sites. So what happens is this cysteine SH is going to accept this acetyl coenzyme A. This pan SH is going to accept this malonyl coenzyme A. There is transfer, decarboxylation, and it rotates around the entire cycle, and when it comes out it is 2 carbon atom more. So suppose if we have to form palmitic acid, this will undergo 7 cycles. 7 cycles in this fatty acid synthase vicinity to form 16 carbon compound, that is palmitic acid. And that is what exactly happens here. Now the question is, which of the following is not a part of fatty acid synthase complex? Ketoacyl reductase, it is present, reduction occurs. Enoyl reductase, enoyl reduction occurs. Acetyl coenzyme A carboxylase is the answer. Rest all the 3 enzymes are a part of fatty acid synthase complex. Acetyl coenzyme A carboxylase is a rate limiting enzyme which is basically the first step of fatty acid synthesis, which is forming malonyl CoA from acetyl coenzyme A. So the answer is C. Then question number 86, carbon atoms added in fatty acid synthesis. Now this is again same what we have discussed in the previous question. You can see, just now I told you ki added in fatty acid synthesis, acetyl coenzyme A has 2 carbon atoms, malonyl CoA has 2 carbon atoms. There is degradation of this, carboxylation is occurring. So 2 plus 2, 4. So we can say 4 in first cycle, and obviously after that subsequently malonyl CoA will be added. So 2 in second cycle. So the answer to this is the carbon atoms added in fatty acid synthesis 4 in first cycle and 2 in the second cycle. Formation of malonyl CoA, then further addition of it. What is the biochemical defect in the Jiggler syndrome? Now this is basically due to the deficiency of the peroxisomes. All of us know that peroxisomes are the site for oxidation of the long chain fatty acids. So isme we can see a long forehead is there, and the child gives an appearance of a Mongolian, which is a characteristic feature of peroxisomal biogenesis disorder. Then question number 88, after digestion by restriction endonuclease, DNA strands can be joined again by. Now see, restriction endonucleases are also known as the molecular scissors of recombinant DNA technology. They always cut at the specific sites. Jैसे EcoR1 is there, PALM-H3 is there, HindH3 is there. There are so many restriction endonucleases which are coming in the market. They can cut DNA into cohesive ends or the blunt ends. Normally we take cohesive ends as one of the very good strands for recombining DNA because the level of attraction, cohesion is more. Now let's look at the enzymes. DNA polymerase. It is clear from the word itself. Polymerase means it is polymerizing, catalyzing the polymerization of DNA. So this is an example of enzyme of DNA replication. DNA ligase. Ligase is a sealing up enzyme. It helps in sealing up the phosphodiester bonds. So whenever DNA undergoes repair mechanism, whenever there is, you can say, breaking of strands of DNA, DNA ligase acts as a joining up, sealing up enzyme. DNA topoisomerases. Topoisomerases, which is catalyzing the topological properties of DNA. It is also known as a stress relieving enzyme. Because we know that DNA is highly intertwined. Whenever we try to break these strands, whenever we try to pull these strands apart, what will happen is there will be breakage of the bonds, there will be denaturation. So this topoisomerases helps in cutting and rejoining the DNA strands. So they are the relieving and stress relieving enzymes of DNA replication. Topoisomerases is of two types, type 1 and type 2. Type 1, if we see, type 1 will cut one strand at a time and type 2 will cut both the strands. And DNA gyrase is a basically what it is present in prokaryotes, so it is helping in unwinding of the helix. So the answer here is DNA ligase, which is also known as a sealing up enzyme. Ligase is a sealing up enzyme. Let's switch over to question number 89. The stored TAGs are hydrolyzed by. Now see, if we look at pancreatic lipase will hydrolyze what? Pancreatic lipase is going to hydrolyze only the dietary TGs. Second is lipoprotein lipase. It is going to digest or degrade only the lipoprotein associated TGs. Lysosomal will degrade TGs in the lysosomes, and hormone sensitive lipase is going to degrade those TGs which are already stored in the adipose tissue. Why? Because this HSL, hormone sensitive lipase, is present in the adipose tissue. It acts here only. So it is going to hydrolyze the stored TGs. So the answer is hormone sensitive lipase. Then a child present in the first week of life with recurrent vomiting, failure to thrive on examination, enlarged liver and spleen were palpated, and blood work, hyperlipidemia and deranged LFTs. So if we see, this is nothing but again calcification of the adrenal glands is there. Hyperlipidemia is there because there is increase in the concentration of TGs, which is again a characteristic feature of Wolman's disease. And the deficient enzyme here is cholesterol ester hydrolase. Right? Then question number 91. Pick the correct pathway of hemoglobin degradation from the given below option. Now we know heme is synthesized. Similarly, it is degraded also. Degradative product of heme is biliverdin, which degrades in the form of presence of biliverdin reductase to bilirubin. This bilirubin binds with albumin, enters the liver, where it undergoes glucouronidation, forming bilirubin, glucourinoid, and this bilirubin then will be converted into urobilinogen and then finally to stracobilinogen. Urobilin, it gives a characteristic color to the urine, and stracobilinogen gives characteristic color to the feces. So let's see where this order is there. Biliverdin, bilirubin, stracobilinogen. So answer to this question is B, where you can see heme, biliverdin, bilirubin, urobilinogen is synthesized before stracobilinogen. Question number 92. Uronic acid pathway is not involved in. Now see, uronic acid pathway is basically uronic acid pathway is the alternative method of oxidation of glucose. Uronic acid pathway. Alternative method. Why? Because the significance of this uronic acid pathway is it gives rise to glucouronic acid. Uronic acid, which plays a very important role in the synthesis of GAGs, glycosaminoglycans or heteropolysaccharides. This glucouronic acid plays a very important role in degradation of heme. It plays a very important role in detoxification mechanism, or which is also known as biotransformation. Now, except human beings, what happens is this glucouronic acid is further converted into vitamin C, which is ascorbic acid. But in human beings lack L-gulunolactone oxidase. L-gulunolactone oxidase. That is why this vitamin C has to be supplied from outside diet. It is a vitamin which is taken in the diet as a supplement. Or the main feature of this uronic acid pathway is generating this glucouronic acid in human beings. Now let's look at the options. Uronic acid pathway is not involved in conjugation of bilirubin. It is involved. Enzyme is glucouronyl transferase. GAGs, glycosaminoglycans or mucopolysaccharides, yes, it is involved. Vitamin C synthesis, no. Why it is not involved in vitamin synthesis? Due to the deficiency of enzyme L-gulunolactone oxidase. And fourth is biotransformation. Biotransformation is nothing but transformation of toxic substances into non-toxic ones, so that they can be excreted as such in urine. Or this is basically detoxification. So bilirubin or you can say glucouronic acid plays a very important role in biotransformation by hydroxylation, by conjugation, as in you have seen in the synthesis of heme it is doing that. Question number 93. A 13-month-old child presented with developmental delay. Her peripheral smear shows macrocystic anemia, no B12 or folic acid deficiency. The compound given in the image is excreted in urine, and her peripheral smear is also given. What is the diagnosis? Now you see, this is nothing but the structure of orotic acid, and the answer would be orotic aciduria. This is basically, and you can see the smear also. So by looking at both the slides, by looking at both the pictures, we can say the person is suffering from orotic aciduria, which is a disorder of pyrimidine metabolism. Orotic aciduria. Basically what happens is pyrimidines synthesis. This is a very important reaction. What happens is carbon dioxide combines in the presence of glutamine. The enzyme is CPS2, carbamoyl phosphate synthase 2, which is a cytosolic enzyme, forming carbamoyl aspartate and finally forming this orotic acid. This orotic acid will form OMP and this OMP will form UMP. Deficiency of orotate phosphoribosyl transferase will lead to excretion of this orotic acid in urine, leading to orotic aciduria. And this is an enzyme of what? Disorder of pyrimidine metabolism. So the answer here is orotic aciduria. And the enzyme is orotate phosphoribosyl transferase. Next question. ApoB48 and ApoA100 is synthesized from mRNA. Difference between them is due to. Now see, these are the only two apoproteins, ApoB100 and ApoB48. These are the only two apoproteins which are basically synthesized by RNA editing. or RNA processing. Now basically what is happening is the synthesis of this ApoB100 takes place in the liver, ApoB100, and in the intestine, ApoB48. It is being synthesized in a normal process, but what is happening is it is coded by CAA. It is coded by CAA. But here what happens is in this ApoB48, this C is replaced by U. The enzyme is cytidine deaminase. Cytidine deaminase means deamination of cytosine is occurring, or it is replaced by uracil. And this UAA is a non-stop codon, or it is a termination codon. So what happens is only ApoB48 is synthesized and it is left as such, whereas ApoB100 is continuously being synthesized by this in the liver. ApoB48 is a very, very important apoprotein for chylomicrons, and ApoB100 is for VLDL. So here the answer would be what? Deamination of cytidine to uridine, conversion of CAA to UAA, which is a termination codon, and the translation will immediately stop there. Then question number 95, true about ribozymes. Ribozymes, again RNA has a catalytic activity. Peptidyl transferase activity, yes. Cut DNA at specific site, no. Participate in DNA synthesis, no. GTPase activity, no. So it is what? It is nothing but peptidyl transferase activity, which is basically peptidyl transferase activity is seen in peptidyl transferase is seen in the elongation step of translation. Where you can see this is the smaller unit of the ribosome, this is the bigger unit, this is the messenger RNA, and here transfer RNA is there. One amino acid it is carrying and the another transfer RNA. So there is formation of peptide bond and then translocation, so that the first amino acid, which is red, methionine, it always stays at the bottom. So the answer here is A. Next question. Triple DNA is due to. Now triple DNA, this is simple Hooke-Steen pairing. Basically this is a non-canonical structure of DNA. What happens here is Hooke-Steen pairing. The triple helix or the third strand of DNA basically forms a hydrogen bond between the major grooves. Major groove of bDNA. This is a non-canonical structure and the pairing is basically known as Hooke-Steen pairing. The third strand forming a hydrogen bond with the major groove of bDNA. There is another fourth strand also, but it should be rich in guanosine residues. But it will comprise of obviously the third strand also. Next question, identify the deficiency disorders. Isme you can see, now see both the pictures, there are rashes, the pustules, the rashes are full of erythema. So this is a characteristic feature of zinc deficiency. Acrodermatitis, alopecia, diarrhea, these are the two very, very important biochemical manifestations. So the answer here is deficiency of zinc, leading to rashes which become red in color. The disorder is acrodermatitis enteropathica. Question number 98. Match the following enzymes with their respective cofactors or coenzymes. Now what are cofactors? Cofactors are basically the non-prosthetic group which is attached to the enzyme. When a cofactor is attached, it becomes a holoenzyme, and just the enzyme part is known as the apoenzyme. Now cofactors are basically helping the enzyme in their reaction. Now see, cytochrome oxidase. Cytochrome oxidase is an enzyme of cytochrome oxidase is an enzyme of ETC, and it requires iron as well as it requires copper for its function. Copper plus and 2 plus. Copper exists in 2 states. Iron exists in ferrous and ferric. Another enzyme is carbonic anhydrase. Carbonic anhydrase requires zinc. Carbonic anhydrase requires zinc. Hexokinase, wherever kinase activity is there, means attachment of inorganic phosphate is there, the enzyme the cofactor is magnesium always, whether it is glucokinase, whether it is hexokinase, whether it is phosphokinase. And fourth is superoxide dismutase. Superoxide dismutase also requires copper for its action. Then question number 99, which of the following are sphingolipidosis? Now see, Tay-Sachs disease. Tay-Sachs disease is caused due to hexosaminidase A. Sandoff's disease, again it is a sphingolipidosis, hexosaminidase B. Krebs disease, again galactocerebrosidase. Fabry's disease, again galactocerebrosidase. One is alpha, another is beta. Wolman's disease does not come under sphingolipidosis. Wolman's disease is basically due to the deficiency of enzyme cholesterol ester hydrolase. So the answer to this question is E. Wolman's disease is not a part of sphingolipidosis. Then last question is, Ehrlich aldehyde test is used for the detection of. Now this aldehyde test is used for the detection of urobilinogen and profobilinogen. So the answer would be 1 and 3 are correct. So here we finish up with the subject wise test series discussion of biochemistry. I hope many of the queries of you people will be solved. So all the best, take care, bye bye.